Hi everyone, this is Mr. N. So I miss you guys. Today I'm going to talk about content related problems and some issues that brought during my office hour, brought by you during my office hours. So have your textbooks ready and go to the page 292 and concept check 15.2. So pause the video, go to the page 292 and concept check question number 2. Okay, so hopefully you paused the video and you have your textbook ready. Question is asking, neither Tim nor Rhoda has Duchenne muscular dystrophy, but their firstborn son does have it. Okay, so we have two people as parents, Tim and Rhoda. I made the pedigree for that. Well, you don't have to, but to show it better and you have an idea how it's gonna work. Tim, circle as male and square for Rhoda as female. And it says Tim and Rhoda do not have problem, but son has it. Alright? So, remember, when we say son has problem, Duchenne muscular dystrophy is X-linked recessive related pattern. So it means that sex chromosomes are affected, specifically X chromosome. When they mention, the question mentions son has it, remember what does son have as sex chromosome? X and Y, correct? Where does the son gets Y? From father. But X has a problem where the son gets the X from mother. So part of the puzzle is solved. Mother is the one who has the recessive gene. So mother is carrier. What does that mean? Mother does not show the problem but has heterozygous, genetic material which shows the phenotype will not be affected, but genotype has one allele as recessively affected. Okay. So let's write down some material. I prefer, there are different methods and strategies some teachers use. I prefer for X-linked or sex-linked diseases. Specifically in AP Biology, we talk about X-linked, not Y-linked. So X-linked diseases, the pedigree, when we talk about, I prefer to write the X and Y to make it clear and easier. So Tim is XY and Rhoda is XX and son is XY. Okay? And so it's recessively inherited. So does Tim have the recessive allele? Of course not. If it has it, it's going to show it because Y does not have the allele, the dominant allele to mask it, right? So XY, Tim does not have that problem. XX for mother, for Rhoda, should have the recessive allele. Can I put another lowercase d for that? For another chromosome? No, I can't. If I do that, it's going to show the recessive character. So I cannot put, I don't know, I said recessive. So I cannot put another recessive d, lowercase d for that. Because based on the questions, we know that mother does not have the phenotype. She is just carrier. She carries the gene. That's why the son has it. The son can get recessively inherited problem from mother because son has X and X does not come from father, it comes from mother, right? So it should be capital D for mother. Based on the son's phenotype, we found out what's going to be the genotype of both parents. Okay? Alright, now it's very easy. You can go for Punnett square to solve the rest of the problem. Let me go to the next page. Okay, so what is the rest of the problem? It says, what is the probability that a second child of this couple will have the disease? Remember, when they say the probability of the second child have the disease, the second child's probability is independent from the first child, unless they say something else. For example, they say what is the probability they have two children and one of them is this and another one is this, to make them together. But in this case, it says one is the probability of second child have the disease. Let me read it again. Child will have the disease, right? For example, let me make it clear. If a family have the first child as son, it doesn't guarantee the next one is going to be girl. All right, they're independent. It's 50-50 chance. The same is the case for this one. So the second the second child's phenotype and genotype is independent of this first one. So we found out that this is the genotype of parents. Father does not show the disease. The phenotype does not the phenotype of the Duchenne muscular dystrophy, so it does not have recessive allele. If father has just one recessive allele, will definitely show the phenotype, but he does not have it because Y doesn't have anything to mask the X-linked related genes or alleles. All right. Mother does not have the phenotype because she does not have two lowercase d's. So this is the genotype. Based on that, we can find what gametes we will have. For father, you can separate gametes, X capital D and Y. Y doesn't have anything related to muscular dystrophy. And mother has one normal gene and one recessive gene. All right. Now let's do the Punnett square. You can pause the video and do it yourself. Normally, we put the male gametes on the top and female on the side. So for father is this one, X capital D, and Y. For mother is X capital D and X lowercase d. And you get these crosses. I'm sure all of you know how to do that. So let's go for the question itself. Question is asking, what is the probability that the second child of this couple will have the disease? When they say child, could be son or daughter, boy or girl, right? So take a look. How many people have the disease? You can pause the video. Definitely some of you will say the probability is 1/2. You may say this one has it and this one has it. Mistake. This one has it, that's correct, because there is no capital D to cover the problem. But this one is heterozygous, and heterozygous will not show the recessive phenotype. It should be two lower cases. Is that clear? Okay. So the answer is 1/4. Children, the child has the probability is 1/4 of 20 or 25%. All right. Okay, and at the end it's asking what is the probability if the second child is a boy. Sorry. What if if the boy has it? It's gonna be again the same because it's gonna be 1/4. What is the probability if the second child is the girl? Well, the girl will not get the problem. 0. For girl is 0. Again, do not make this mistake. It's recessively inherited, problem. All right. And for being recessively the phenotype to be expressed, you need two lowercase alleles. So this one it does not show the problem. The probability is 0. Okay. Now let's go to another concept. Page 295. Basically, it starts from 294, recombination of linked genes, and 295. Recombination of the linked genes. So You remember the crossing over happens in meiosis 1, prophase 1, right? What is the role for that? It's just like mix some alleles together to make a new combination, the recombinant gametes. So you may have the same gametes as parental gametes, and you get some mixture of them, right? That's the role for crossing over, recombination, new types of gametes. Okay, for simplicity, these two are homologous chromosomes, and crossing over happens between, well, I should have made them closer together. For example, let me do it here. So it should happen between non-sister chromosomes, okay? Not sister, non-sister chromosomes. All right. Imagine we're, considering dihybrid recombination. I mean dihybrid cross. So you have two homologous. Well, basically, when we show the chromosomes, it's the replicated one. So you have two copies. For simplicity, we can get rid of one copy and have these together to see what's going to happen during crossing over. All right. Let's go to the next page. Here. So you have two genes. Gene A is located here, the capital allele is here, lowercase allele in another chromosome is here. Remember, there used to be X. I got rid of one copy to make it simple to show you, like an interphase chromosome that they have not made the duplicate. Anyways, A and B, right? Lowercase a, lowercase b. So crossing over just like exchanges two alleles together. For example, for this one, if crossing over happens, for example, here, these two segments to just like get switched. Here you will have B, and here you will have, capital B. It got switched, right? So now you have new types of gametes. If you remember, I used to do something very simple. Let me write it down. I'll show you. A, B, B. I used to do like this. So B, B. I switched it. I should have done it earlier. B, B. This is the genotype for organism for two types of genes. Okay, when we're supposed to make the gametes, I used to go like this. The first one from A to B, this is the original gamete. This happens if the crossing over does not happen in between, and just like two genes goes to gamete as exactly like parents. All right, just crossing over would not happen, or the crossing over happens somewhere else, not between them, to switching happen. They both go to the gamete, and gamete will look like parental genotype. So one is AB. Another possibility that I showed already, could be A lowercase b, that we did the crossing over. And here you can have the crossing over one, a capital B, or original one, lowercase a, lowercase b. All right. So two types of gametes are possible: parental-like gametes and recombinant ones. All right. So there are enough distances between two genes, two different alleles of two genes. But what if, let me draw here. What if A is, and here is the A, what if A and B and the corresponding alleles on another chromosome are very close to each other, there are a lot of possibilities crossing over happens somewhere else, and these two, they go to the gametes. Mostly they go to the gametes that make the gametes like parental gamete. All right. So the possibility that something crossing over happens in between still exists, but it's very, very less. So most of the gametes you get, they're like parental gametes. All right. So based on this philosophy, based on this kind of explanation, scientists came up with the formula of recombination frequency. Those genes, they're very close to each other. They're called linked genes. Okay, they're linked genes because they're so close to each other. They can go to the possibility that they go to gametes are a lot. So based on the recombination frequency, you can based on the result, you can calculate the recombination frequency. For example, if the number of the recombinant gametes are just like a lot, like normal Mendelian based crossing over outcome, just like 2 gametes are parental gametes, 2 are recombinant, it's just like 50-50 between each. But if they're very close to each other, the recombinant number will be lower. When the top part is lower, the result will be a smaller number. So recombination frequency, if the number is very low, is a small, shows the genes are very close to each other. Does it make sense? If the genes are far apart from each other, the possibility that the recombination happens is a lot. I mean, it makes a final recombinant genes, recombinant gametes, and recombinant parents 2 and 2, 50-50. So 2 possible gametes for parental style and 2 possible for recombinant ones. So this is my explanation about these 2 topics. If you want to read more for the linked gene, you got to read the 295, 294, and 95. So work on it, read one more time, and prepare yourself. So sex-linked, not the recombination, is going to be in your Friday test. If you have any question, my office hour, email, just like I'm one typing away from you. So send it to me. I'm going to have more videos that I can elaborate on the content related problems.